Hello everyone, myself Dr. Sunaina Rathi, instructor for the course Biochemistry. Through this video, we are going to demonstrate the qualitative tests for carbohydrates, amino acids and proteins. So first, we are going for qualitative tests of carbohydrates. So the aim of the qualitative test for carbohydrates are to study the properties of carbohydrates and to identify unknown carbohydrates based on certain color reactions. So the following tests are a few of them performed for qualitative determination of carbohydrates. First is Molisch test, Fehling's test, Barfoed's test, Seliwanoff's test, Iodine test and Hydrolysis test. Molisch test. So this test is named after Austrian botanist Hans Molisch, and the principle of this test is that in presence of concentrated sulfuric acid, the carbohydrates will be dehydrated to furfural and hydroxymethyl furfural, which will condense with two molecules of alpha naphthol to form violet or purple colored complex, and this is a general test for detection of carbohydrates. Disaccharides or polysaccharides also give this test because they will be first hydrolyzed by the concentrated acid to monosaccharides, which will be converted to 5-hydroxymethyl furfural or furfural derivatives, and they will react with alpha naphthol to give violet or purple colored complex. First we are going for Molisch test. So this is Molisch reagent. Now we will take 2 ml of test solution and we will add 500 microliter of Molisch reagent to it, and we will mix well. Then we will keep the tube in the slanting position, and carefully from the side of the tube we will put about 1 ml of concentrated sulfuric acid without mixing. And a red cum violet ring will appear at the junction of the two liquids. So this red cum violet ring which appears at the junction of the two liquids indicates the presence of carbohydrate in it. Fehling's test. So Fehling's test was developed by German chemist Hermann von Fehling in 1849. So sugars with potentially free aldehyde or ketonic group will be able to reduce the metal ions under alkaline condition, and this reducing sugars will produce indiol in alkaline solution. So these indiol forms of sugars in alkaline solution, they are very powerful reducing agents, so they will be able to reduce oxidizing ions such as copper, and the sugars will be oxidized to complex mixture of acids. Reducing sugars in presence of alkaline condition reacts with metal ions such as copper sulfate to produce initially a yellow colored compound called copper hydroxide. Then when the solution is heated, it will produce copper oxide, which is precipitated as red precipitate, and this red precipitate or the rusty brown precipitate will indicate the presence of reducing sugar in it. Next we are going for Fehling's test. So this is Fehling's reagent A and Fehling's reagent B. So now we take equal volume of Fehling solution A and Fehling solution B in a test tube, and we will mix it. To this, we will add few drops of test solution and then we will boil in a water bath for 10 minutes. After boiling in the water bath for 10 minutes, rusty brown or red precipitate is observed, which indicates the presence of reducing sugar in it. Next is Barfoed's test. Barfoed's test was developed by Danish chemist Christian Thomsen Barfoed. Here, the monosaccharides present in the test solution, they will react with cupric ions and the cupric ions will be reduced to cuprous ion, which will give a brick red color precipitate, and this big red precipitate will indicate the presence of monosaccharides in it. So this is Barfoed's reagent. So first we will take 2 ml of Barfoed's reagent and in it we will add 1 ml of test solution and we will slightly mix it and then we will boil for few minutes in a water bath and we will cool it. So there is a formation of brick red precipitate, and this formation of brick red precipitate indicates the presence of monosaccharides in it. So next is Seliwanoff's test. So this test is named after Theodor Seliwanoff. So here the ketoses are dehydrated more rapidly than aldoses to give furfural derivatives by acid, which will be then condensed with 2 molecules of resorcinol to form red colored complex, and this red colored complex indicates the presence of ketose sugar. This is Seliwanoff's reagent. So first we will take 2 ml of Seliwanoff's reagent and there we will put 1 ml of test solution and then we will mix it slightly. Next we will heat this mixture just to boiling, and the appearance of deep red color will indicate the presence of ketose in it. Iodine test. So the iodine starch test was first described by J.J. Collin and H.F. Gaultier de Clobbery and independently by F. Strangier in 1814. The interaction between starch and the triiodide anion is the basis for iodometry. So the amylose fraction of starch and iodine form an inclusion complex where the linear amylose molecule will coil into a spiral in solution, which is held by hydrogen bonding between hydroxyl groups of adjacent loops, and the iodine molecules align themselves in the center of the tube formed by the spiral. So this arrangement allows the interaction of the electronic orbital of many iodine atoms, which cause the light absorption that gives the complex its characteristics deep blue color. So starch forms a very dark blue-black complex with triiodide. However, the complex is not formed if only iodine or only iodide is present. The color of the starch complex is so deep that it can be detected visually when the concentration of the iodine is as low as 20 micromolar at 20 degrees centigrade. Iodine test. So this is iodine reagent. For this we will have to take 1 ml of test solution and we will add few drops of iodine solution in it, and the appearance of deep blue color indicate the presence of starch in it. Next is hydrolysis test. So sucrose and starch can also give positive result with Fehling's test after hydrolysis. So all oligosaccharides and polysaccharides, they are converted to monosaccharides when they are exposed to concentrated acid, and the monosaccharides will give positive result for Fehling's test. So the common example is sucrose and starch. For this, we will take 1 ml of test solution and add few drops of concentrated HCl, and we will boil the mixture for few minutes and we will cool it down. Next, we will neutralize it by adding sodium hydroxide, and we can check it by litmus paper. Then the solution will be tested with Fehling's reagent, and we will get rusty brown or red precipitate, which will indicate the presence of reducing sugar in it. Next is qualitative tests for amino acids and proteins. The aim of the qualitative test for protein and amino acid is to identify unknown amino acids based on certain color reactions. So through this video, we are going to demonstrate how to perform qualitative estimation of amino acids and proteins. So we will be performing 4 different types of tests to estimate amino acids and proteins. First is Ninhydrin test. So this is a general test for detection for both amino acids and peptides. Second, Xanthoproteic test. So this is to identify aromatic amino acids. Third is Millon's test. So this test identifies the presence of tyrosine in it. And fourth is a Biuret test, which is a general test to identify proteins and peptides. Ninhydrin test. This test is for detection of primary and secondary amines or amino acids. Here, a ninhydrin molecule initially reacts with a free amino acid by deamination and decarboxylation of the amino acid at an elevated temperature to produce reduced ninhydrin, and the reduced ninhydrin will react with the second molecule of ninhydrin to produce a blue colored or a violet blue condensation product known as Ruhmann's purple. This violet or purple color indicates the presence of amino acids or peptides. Amino acids like proline and hydroxyproline also react with ninhydrin, but to produce a yellow colored instead of a purple colored complex. Ninhydrin test. To 2 ml of test solution, add 1 ml of freshly prepared 0.1% ninhydrin solution. Mix the contents and boil for a couple of minutes. Allow it to cool. So, the violet or the purple color which is observed indicates the presence of amino acids or peptides in it. Next is Xanthoproteic test. This test is used to detect amino acids containing an aromatic nucleus like tyrosine, tryptophan or phenylalanine. So, the benzene ring in the amino acids will be nitrated by heating with nitric acid, and it will form yellow nitro compound which will turn to orange color in presence of alkali. The orange yellow color will indicate the presence of free aromatic amino acids. Xanthoproteic test. To 2 ml of test solution, add 2 ml of concentrated nitric acid and boil the content, and after cooling add excess of 40% sodium hydroxide. The yellow to orange color which is developed indicates the presence of free aromatic amino acids. Next is Millon's test. This test is specific for identification of tyrosine. So here protein is denatured by mercuric sulfate exposing hydroxyphenyl group of tyrosine. Then sodium nitrite reacts with sulfuric acid to form nitrous acid. Then the exposed hydroxyphenyl group will react with the nitrous acid and the compound which is formed will chelate the mercuric ions and give red color precipitate. This brick red color precipitate will indicate the presence of tyrosine. Millon's test. To 1 ml of test solution, add 1 ml of Millon's reagent and then boil gently for 5 minutes. Cool under a tap water and add a few drops of 1% sodium nitrite solution and warm again gently, and the red color develops indicate the presence of tyrosine in it. Next is Biuret test. So this test is positive for compounds containing 2 or more peptide bonds. The name came from Biuret, which also gives positive result since all proteins and peptides possess at least 2 peptide linkages. Thus, a tripeptide can give positive Biuret test. The test is negative for amino acids which do not contain a peptide linkage or for dipeptides which contain only one peptide linkage. The principle of Biuret test is that the alkaline copper sulfate will react with compounds containing 2 or more peptide bonds to give a violet colored product, which is due to the formation of coordination complex of cupric ions with unshared electron pairs of peptide nitrogen. So this violet color or deep blue color developed indicates the presence of protein or polypeptide. Biuret test. To 1 ml of test solution, add 1 ml of 10% sodium hydroxide, mix well and then add few drops of 0.1% copper sulfate solution. The violet color develops indicate the presence of protein or polypeptides in it. Thanks for watching the video, and here is the email address for further feedback and queries. Thank you all. Namaskar.